Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing the Gujarat SET question paper for the subject Life Science. This is paper 2 that I am discussing. It was conducted in the year 2024. Now coming to the next question, so we have guanosine triphosphate or GTP hydrolysis is required by which one or more of the following steps in protein synthesis. So first one, they have told activation of amino acid by amino acyl tRNA synthetase, it requires ATP. So here is your amino acid plus ATP. In the presence of amino acyl tRNA synthetase, you will get amino acid linked to AMP, amino acid AMP, which is the intermediate, and an uncharged tRNA will enter there, and this amino acid will be transferred from the AMP intermediate to the 3' end of the tRNA, and you will get amino acyl tRNA and AMP will be released. So here it is ATP, not GTP. Then attachment of ribosome to endoplasmic reticulum also doesn't require GTP. Then translocation of tRNA nascent protein complex from A site to P site. So EFG, elongation factor G, is involved in the translocation. So here you have your nascent polypeptide attached to its tRNA at the A site. P site is without any protein or amino acid. Now the ribosome has to move from this P site to E site and from A site to P site, so that A site becomes free for the newly amino acylated tRNA to enter there with a new amino acid. So for that you use the elongation factor EFG, which is bound to GTP. Now the GTPase on the ribosome will cleave this EFG GTP to GDP plus PI, and that energy will move the ribosome and tRNA from to the next binding site, so that the tRNA which is uncharged and present on the P site move to the E site and is removed off, and the tRNA with the polypeptide attached on the A site moves to the P site, and the A site will be now free for the incoming tRNA molecule. So here it is GTP that is involved, not GDP. So R is one answer. Okay, that requires GTP. Then next one, binding of amino acyl tRNA to A site. Now that is with regard to your EFTUTS cycle for binding amino acylated tRNAs. So here in the beginning, so you have the F-met alanine methionine attached to that, alanine was attached, and now the A site is free, the ribosome has translocated, and the charged tRNA has to bind to the A site. So here your charged tRNA is the one with serine. So it will be bound by the EFTU GTP complex, and it will enter into the A site. After binding, the GTPase activity will cleave off the GTP bound EFTU to EFTU GDP, and EFTU will dissociate from the amino acyl tRNA and as well as from the ribosome. Now the EFTU bound GDP will be bound by the EFTS. Upon EFTS binding, the GDP will be dissociated off, and the then The new GTP will bind to the EFTU EFTS complex, and the EFTS will dissociate upon binding of GTP to EFTU, and the EFTU GTP is now available for binding to another amino acid tRNA. So S is also correct. So your answer is option D, that is R and S only. These are the cycles which require GTP. Then next one, the maximum buffering capacity at a physiologic pH would be provided by a protein rich in histidine. Okay. Histidine has a pKa value near to physiologic pH. Then next one, in scurvy, which of the following amino acid that is normally part of collagen is not synthesized? Now collagen, it is a triple helix, and it has repeating units of glycine, proline, and 4-hydroxyproline. Now scurvy, it is due to the deficiency of vitamin C. And vitamin C is a cofactor of proline hydroxylases. Proline hydroxylases are enzymes which attach a hydroxyl group to proline. Now in scurvy, vitamin C is not available in sufficient amount. So the enzyme proline hydroxylase cannot function properly. It will not be able to convert proline to hydroxyproline. So which of the following amino acid normally part of collagen is not synthesized? It is hydroxyproline. Okay, so the answer is option D. Now next one, a researcher has to isolate a lysosomal membrane protein that recognize mannose 6-phosphate group in lysosomes. Which of the following chromatographic techniques for the purification of protein could be used to specifically isolate this particular protein of interest? So here it will be good to use affinity chromatography, because affinity chromatography works on the principle of the protein receptor binding to its ligand. So here your protein receptor will be the one which will bind to mannose 6-phosphate groups in the lysosome. So if you have such a receptor, attach that receptor to the affinity column. Your ligand, that is lysosomes with the mannose 6-phosphate group, will go and bind there based on the principle of protein ligand binding, and you can purify. So affinity chromatography will be the answer. Then next one, which motif in a protein strongly suggests that it is a DNA binding regulatory protein? Now if the protein is having motifs like a helix turn helix, leucine zipper and zinc finger, then it suggests that it is a DNA binding regulatory protein. So here in this option they have given only zinc finger, so the answer is option D. Now next one, contraction of cardiac and skeletal muscle is initiated by the binding of calcium to which of the following? So when the cardiac muscle is stimulated, calcium ions are released from your sarcoplasmic reticulum. In the cell cytoplasm, calcium ion concentration increases. This calcium, it will go and bind to troponin and change its conformation. This in turn, it will move the tropomyosin away from the active site of the actin filament. Now this active site can bind to myosin heads and result in contraction. So contraction of cardiac and skeletal muscle is initiated by binding of calcium to which of the following? It is troponin C or troponin as in given options, that is answer D. Then next one, which of the following is the important reactive group of glutathione in its role as an antioxidant? It is the sulfur hydryl or the SH group. Now this group, it can donate its hydrogen atom to free radicals and neutralize them and protect the cell from oxidative damage. Then next one, warfarin acts primarily as. Now there are certain coagulation factors, that is 2, 7, 9 and 10, which require the gamma carboxylation by vitamin K for them to have normal biological activity. Okay. Now vitamin K, it is obtained from food sources and it will be reduced to vitamin KH2 by vitamin K reductase. Now this vitamin KH2, it will be oxidized to vitamin KO and it is coupled to the gamma carboxylation of these coagulation factors. So carboxylation occurs at the glutamic acid residue. So here you can see there is no carbon dioxide here, but here you have one over here. So this oxidation of vitamin KH2 to vitamin KO is coupled to the gamma carboxylation of these coagulation factors. And then this vitamin KO is reduced to vitamin K1 in a reaction catalyzed by KO reductase, and again the cycle continues. Now what does this warfarin do? Warfarin, it will inhibit the KO reductase, thereby blocking the formation of vitamin K1 from vitamin KO. Now vitamin K1 is blocked means vitamin KH2 is also blocked. Now vitamin KH2 blocked means it cannot be oxidized back to vitamin KO. Now no reaction of conversion of vitamin KH2 to vitamin KO means there is no gamma carboxylation of your coagulation factors. So coagulation factors will have very less biological activity. So warfarin act primarily as an anticoagulant. Okay. So the answer is option B. Then next one, a microorganism that uses inorganic compound as source of energy and fixes carbon dioxide is best defined as. So in this case you have to remember this table. So if you see the table, you can see photoautotroph. So photoautotroph, they use light as energy source, so that cannot be the answer. They have given inorganic compound as source of energy. Chemoheterotroph, okay, they are using organic compound as source of energy, not inorganic. Chemolithoautotroph, yes, they are using inorganic compound as energy source, and inorganic compound as electron donor, and fixes carbon dioxide. So carbon dioxide is the carbon source. Okay, so chemolithoautotroph will be answer. Chemoorganotroph means they are using organic carbon source, not carbon dioxide. So not chemoorganotroph. So the answer is chemolithoautotroph. So this table you can write down for study. It is very easy to remember this one. See here, all photo means light is the source of energy. Chemo means either organic or inorganic compounds are the source of energy. The only thing that is changing everywhere is your electron donor and carbon source. But in majority of the cases it is carbon dioxide which is the carbon source. Oh no, it is almost equal to your organic compound also as carbon source. Okay. So that's it with regard to this question, and come to the next one. You can pause the video and write this down. Okay, then which of the following is true about metagenomics? So metagenomic, it involves studying the genome of the entire microbial community, which you get from the bulk of the environmental sample. There is no isolation, there is no culturing of the various microorganisms that you obtain from the sample, nothing like that. You use the whole environmental sample and isolate DNA and study. And it provides information based on phylogeny, and functional information also is obtained. So what they have asked, it is the analysis of genome of entire community. Yes, not individual organism. It doesn't require isolation, cultivation. It gives phylogenetic information but not functional. No, it gives both. Both phylogenetic and functional information is obtained. So B is the answer. Then environment A has 100 species, environment B has 600 species. Therefore, environment B has greater species richness. What is species richness? It refers to the number of different species in an environment. So here environment B is having more different species than A. Now what is this abundance? Abundance means total number of individual organisms in that area. Total number of individual organism in that area. Now if you have a forest with 100 trees, you will say that you have got 10 rubber trees, 20 will be apple trees, and 70 will be your pine trees. So 10 plus 20, 30 plus 70, 100. So that is abundance. Total number of individual organisms. So individual apple tree, individual pine tree, how many are there each? That is abundance. Now what is species evenness? Species evenness means how evenly the individuals of a different species are distributed in a community. It can be high species evenness or low species evenness. High species evenness means each species will have almost a similar number of individuals. That is, if you take a forest, if there are 100 apple trees, you will have approximately 110 pine trees, or approximately you will have 90 some other tree. So that is high evenness. That is, each individual species are almost even. 100, 110, 90. These numbers are approximating to nearly 100. So that is evenness. Species evenness. And here this example what I told, it shows high species evenness. Now low species evenness means there will be 100 apple trees, but you have only around 10 teak trees and 10 pine trees. They are not even. One is 100, other both are 10. How it will be even? They are not even. So it is low evenness. That is what is meant by evenness. Okay. I don't know what is equity. I searched but I couldn't get much information on that. Then next one, a placid cell of water potential minus 0.732, solute potential minus 0.732 and pressure potential 0. Was dropped in a 0.1 molar sucrose solution of water and solute potential equal 0.744. What will be the pressure potential of the cell at equilibrium? Okay, the question is for at equilibrium. Okay, so you know that water moves from an area of high water potential to an area of low water potential. So initially the water potential was minus 0.732, and when it was dropped in a solution, the solution had a water potential of minus 0.244. So here this is at a higher potential than the cell. So water will enter from the solution into the cell till equilibrium is reached. That means both the sides are equal. Okay. So at equilibrium, the osmotic potential of the cell will be equal to that of the solution. So it will come up to around minus 0.244. Now substitute the values. So water potential will be minus 0.244. Solute potential will be this, that is minus 0.732. You have to find the pressure potential. In placid cell there is no pressure, it is 0. Whereas in cell at equilibrium, after it is put in a solution with higher water potential, then turgor pressure will be there, pressure potential will be there, and you can calculate using the formula. Water potential is equal to solute potential plus pressure potential. You will get it as 0.488. So it is either B or D. Here it is hidden there behind this photo. I think it is B. Okay. So it is 0.488 is the answer. It is there in your options, but it is hidden by the photograph I have placed. Then next one, which of the following activities is not present in clino fragment but present in intact DNA polymerase 1? Now when you treat this E. coli DNA polymerase 1 with the protease subtilisine, you will get a large protein fragment that is called as the clino fragment. Okay, and this clino fragment, it is having 5' 3' polymerase activity as well as 3' 5' exonuclease activity or the proofreading activity. Now they have asked which is not present in the clino fragment. So it doesn't have the 5' 3' exonuclease activity present in the DNA polymerase 1. 3' 5' polymerase, no, it is having 5' to 3' polymerase. 3' to 5' exonuclease, yes, that is proofreading activity, it is present in clino fragment. 5' to 3' polymerase, yes, it is present in clino fragment. Then next one, which of the following sequence indicates the correct structural organization of chromosome constituents going from smaller to larger size of molecule? So here you have the DNA, then the DNA is wound around the histones. Then there is the nucleosome, which is the bead of string arrangement. Then the nucleosome condense together to form the solenoid. Then there are the chromatin fibrils which are forming loops. Again they will condense together and you will get the final condensed chromosome. Now see the options. So first chromatin fibril, then nucleosome, no, not correct. Histone octamer, no, histone octamer comes before nucleosome, so that is not the answer. Then nucleosome, chromatin fibril, chromosome loop, histone octamer in the end, no, not correct. Nucleosome and histone octamer, no, histone octamer forms, then only nucleosome forms. So C is also not correct. D, if you see histone octamer, yes, then nucleosome, yes, then chromatin fibril, yes, then chromosome loop, yes. So D is the answer. Then next one, you are given two proteins in two tubes, that is A and B. Protein A is homodimer. Dimer means two subunit. Homo means same subunit. Okay. Same molecular weight for both the subunits. That is homodimer. While B, it is homotetramer. Tetramer means 4 subunit. Homo means all equal molecular weight. They are run on SDS page. Both the gels gave single band at 100 kDa. Okay. So protein A run on SDS page. You homodimer is protein A. You got 100 kDa band. Now protein B run on another SDS page. It also gave a 100 kDa single band. The molecular weight of the protein. Now SDS, it breaks the non-covalent bonds that hold the protein subunits. So individual monomers are separated and they can be analyzed. So homodimer means two subunits. So the monomer here what you got is 100 kDa. So dimer 2 will be there. So 200 kDa. Okay, that is there in option A only. So A must be the answer. Then homotetramer, tetramer is 4 subunit. So one monomer is 100 kDa. So 4 will be 400 kDa. So B will be 400 kDa. So the answer is A. Then next one, under normal measuring conditions, you measure the absorbance of a solution in 1 ml and 3 ml cuvettes. You will expect the absorbance value in. Now absorbance you all know is epsilon Cl. Epsilon coefficient constant. Okay, C is your concentration and L is your path length. Now here what have they changed? They have changed only the size of your cuvette. It is not dependent on any absorbance equation. So what will be the absorbance value? It will be identical in both A and B. Only the cuvette size is changed here. It is not dependent on your absorbance value. So that's it with regard to the set paper. I have discussed the whole question paper for that is paper 2. Thank you for watching. Please do like, share and subscribe. If you want still previous years of Gujarat SET exam, you can go to my channel Omni Bio Entrance. In the channel playlist, see for Gujarat SET exam and you will find them there. Thank you.